class will last a little bit longer after 4 o'clock. We'll need some minutes longer. Before entering into some ideas of the origin of the nervous system, what I will do now is just to finish the Hawks story, and then we'll talk about the differentiation of the somites, and we'll realize that the somites and the nearby neural tube are in close contact, and there is a lot of interactions between those tissues and others, and this would be a kind of bridge before entering into the origin of the nervous system. Just I would like to repeat the information on this slide because I think it is very important for you, for the future, to understand how genes can work. And main point was trying, and I'm not sure that yesterday I explained that properly. So the main idea is try to understand how a combinatory action is working. So we have here a model of genes, and actually they are organized from 3' to 5'. You know that what you have here is the pattern of expression from posterior to anterior, or from anterior to posterior. Okay, so we have the gray, green, and blue, and the posterior is expressed more posteriorly, the anterior in the entire zone here, and this one in the intermediate zone. Okay, so the question is what kind of phenotype do you get with that? In this model, the phenotype will be a combination of each, right? So we have these 3 segments which are going to be gray. These ones is a combination of gray and green, and these ones is a combination of those 3, right? If this is true, if we mutate gene number 2, which is the figure that you have here, now the result, according to a combinatory action or a kind of code in the cells here, this is one possibility. The result is going to be what you have in this picture here. An organism, this is the head, this is the tail, and those segments here are not going to vary. The phenotype is going to be exactly the same as in the wild type, because there is no action of gene green, gene number 2. But the phenotype of the mutant situation, the knockout, the mutant situation is going to be, the phenotype is going to affect to this entire region, right? So this area here, because there is no green gene, now the phenotype of that is going to be exactly the same as the segments in the most anterior part, okay? But the phenotype of this area here is going to be something new. It's going to be something that it does not exist. Here, those 3, according to this model, those 3 segments are the result of the combinatory action of blue, green, and gray. And now here, if the combinatory action is the way that Hawks work, we are going to get a new phenotype here, which is going to be triggered by the expression of blue and gray, okay? Now, there is another possibility, and the other possibility is what we have here. Here we have the same pattern of expression, and the possibility is that there is certain dominance. And the idea is that posterior genes, which mean genes that are more in 5' and are expressed more in the posterior part of the animal, are dominant over the other ones. Do you follow that? Now, what it means that? It means that in those three segments, the expression, the phenotype in normal conditions, there is no mutation here, is going to be gray. In those 3, is going to be green, because green is dominant, 2 is dominant over 1. So it is a little bit more posterior, it is more towards the 5', so it's going to be dominant. And this one is going to be blue. And it's going to be blue. Why? This is going to be blue because it is dominant over the other 2. Now, what is going to happen in the mutant condition? We have a mutation, a loss of function of this gene and gene number 2, and then the phenotype. The phenotype of the mutant. If this model is true, the phenotype is going to affect only in those three segments and not in all those ones. And this is what I wanted to make you think about. Okay. Now, if this is true, the phenotype is going to be affecting only in those three, because now the dominant effect of this green is lost, and now it has to be only gray, right? But here, the phenotype is going to be exactly the same as in the wild type, because blue is dominant over green and gray, and green is not there. Blue is expressed normally. So what we are going to get here is an embryo in which those posterior segments are going to be blue. Those central segments are going to have the same phenotype as the anterior ones. And actually, I have to tell you that this is exactly the truth. This is the way that the system works. And it has been found in mammals that when there is a mutation, this mutation results in a phenotype, in the loss of function mutation, okay? So it means loss of function, the gene is not working. Now, then in that condition, the phenotype is going to be exactly the same phenotype and as they have the segments that are immediately before to that one, okay? And that demonstrates that this model here, which is called the posterior prevalence, which means that as more posterior, they prevail their phenotype over other ones. And it has been demonstrated that this is true. And this is what Mario Capecchi, this Nobel Prize, found, that mutations in those segments here in genes, in three genes, he had to delete all three genes, resulted in a phenotype that was more anterior to the one, which is exactly this model here. That's the way the Hox genes are working. And I wanted you to think a little bit more about that, because this is something that you may find in the future in different instances, in different examples. Now, there are more interesting things about the code of the Hox. I'm not going to enter into detail. We will go back to this. Okay, so the upper half of this slide corresponds to a chick embryo. The lower half corresponds to the mouse embryo. And what we have here, that represent the somites. This represents the vertebra, and this is the mesoderm, the combinatory actions of the different Hox genes. And actually, there are more important things in addition to confer the identity of the different vertebrae and the different somites. For example, and this is something that is conserved in vertebrates. Okay, so the transition from cervical to thoracic to lumbar to sacral or to caudal vertebra is exactly indicated by the same code in the same code, the same combination of Hox in chick as in mouse. And this is what you have here. Okay, so the transition to thoracic and lumbar actually is which is this area here. This is indicated by those Hox here, which are exactly the same Hox as in the chick embryo, and this is conserved in many organisms, from fishes to frogs to birds and to mammals. And then there are other combinations like this one where C5 and C6 encounter. This is the signal where it is indicating to this animal anterior-posterior where the anterior limbs have to be developed, and this happened to you as well. Okay, so the anterior limbs of this chick, which is the wing, is reading those somites. This area here is reading where it is this combination, and here an appendix, a wing, is going to develop. And in the case of the mouse, the anterior leg is going to develop here. In the case of humans, the arm is going to develop in this, exactly in the C5, C6, and for the fins in fishes also is the same combination of Hox that is indicating this story. Okay, now this is something that when we study the limb, I'll go back to this point. I would like you to tell just brief things, so a couple of things related to this the differentiation of the somites. And I would like you to point to this area here. This is the neural tube, the notochord, the neural tube, the notochord, and this is the somite. Here we have the epidermis, which is dorsal. The epidermis originally comes from the ventral part of the ectoderm, but after neurulation and gastrulation, the epidermis is covering the entire animal, right? And we have here this environment, this microenvironment, which is characterized by signals from all those tissues that are going to be very important to stimulate the differentiation, the development is here. So somites, neural tube, and other types of tissues that are here are sending signals, are communicating, are talking to each other in order to generate, particularly the notochord is going to be very important in this process. The notochord is acting as a source of signals that are going to be controlling the states, right? And very soon, somites that are going to be regionalized, are going to be separated in different domains. For example, this area here, which is painted in blue, this area that is more in the ventral side of the somite, and medial means it goes to this line, is a part of the somite that is closer to the notochord, and this part is going to undergo an epithelial to mesenchyme transition, which means that the cells detach and are start to proliferate and migrate. And this is what is called the sclerotome. And the sclerotome is the part of this somite that is going to cover the spinal cord in order to form the vertebra. So the same is happening in the other side. Some cells from this somite and cells from the other somite in the other side are going to construct one vertebra here. Now, there are other processes in it. For example, this ear of the somite is going to the dermamyotome, which is going to be split into different tissues. Okay, the dermatome and the myotome. The dermatome, which is here, is going to develop the dorsal dermis of the animal, and actually is corresponding to the dorsal dermis of your body, which means the dermis of your back. Okay, here we are. Okay, this is the dermatome, and is this area here, right? Now, there is this part here of the dermamyotome is called area here, that then it will split in these two, right? So dermamyotome gives rise to the dermatome and the myotome. The myotome are those cells here that are bending just below the dermatome here, and they are going to develop the muscles of the vertebra, and actually are the muscles that are holding the vertebra. Okay. Now there is another population of cells, which are those cells here that also suffer an epithelial to mesenchyme transition and are migrating, and depending on their different ways of migration, and they will develop different parts of the muscles. For example, the muscles from the body and the muscles of the limbs of your body are coming from those migrating cells from each somite, and they differentiating muscles in different parts. Okay, so here we have this sclerotome that will develop chondrocytes, and the chondrocytes will give rise to vertebra. Depending on the position, anterior-posterior organization, they will develop they will become they will differentiate in vertebra holding some ribs, and also some cells will develop the cartilage or the tendons and so on. Now the migratory cells are these ones here that develop the muscles, and then we have the dermamyotome. There are some other regions in the somite which I don't want to go into it. It's too much detail. Okay. Now I would like you to pay attention to this half of this picture here, which is telling you exactly the same story as we saw here. It is exactly the same, but we are going to put some names to the molecules that are responsible for this regionalization of the somite. For example, I would like to start with a notochord. The notochord is expressing sonic hedgehog, and sonic hedgehog, you know the hedgehog signaling in vertebrates, we call them sonic hedgehog, and sonic hedgehog is going to be very important to target the ventral part of this somite, which are the blue cells here, and to stimulate the differentiation of the chondrocytes of the vertebra. And also sonic hedgehog is going to induce in the ventral part of the neural tube to develop the specific part of this of the spinal cord. And then even some sonic hedgehog of the spinal cord, this part is going to also to be released and is going to influence the differentiation of the chondrocytes. Now, this is driven because downstream of the sonic hedgehog signaling pathway, a transcription factor, which is called PAX1, and correspond to one of those what we call master genes. Okay, master genes are genes that when they are activated, when they are on, they are expressing them, a particular lineage is going to be activated. Okay. Now this is exactly what is happening here. So we have the PAX1 is going to be activated, and this is going to be very important for the differentiation of the chondrocytes. Okay. Now I would like to ask you: is the sound okay today? Is the sound? Could you hear my voice properly? So far, yes, Victor. Thank you. Still, sometimes you have problems. Sometimes, a couple of times has been caught. Okay, I'll see what's happened. Okay, now, right. So the other possibility, now we are going to do one thing. I'm going to switch off the image and see whether for the I'm going to switch off the video and see whether until the end. Yes, I know. This is what they told me, the students coming. I'm going to see whether it's better without video. Okay. Now, in any case, what I was telling you is that the sonic hedgehog is acting in from the notochord as a signaling center. All right. So sonic hedgehog is triggering here, and this is going to start differentiation. There are other signals that are very important. For example, this myotome that is going to develop the muscles that are keeping the vertebra tight to each other. What they do is actually they require low levels of sonic hedgehog because this part is a little bit far away. Low levels are able to reach here from the ventral part of the neural tube or from the notochord, and those low levels of sonic hedgehog plus some wind that is coming from the dorsal part of the neural tube, this is important for the differentiation of the muscle of the myotome. The myotome, I remember you that is this one which is developing the muscles of the vertebra, which is this area here, okay? The myotome, you see it here, okay? Now, so, and the same happens for the dermatome, for the dermis, which is coming from signals from the epidermis and also from the dorsal part of the neural tube, and some wind from the epidermis, and some signals from ventral mesoderm, like BMP4, which is very typical from the ventral mesoderm. You know that, okay? This is going to be very important here for triggering the differentiation of those migratory cells that are going to develop most of the muscles of your body and your limb, okay? Now, let me tell you a few things. I would like you to have a very quick picture of what is happening during differentiation. So what we have seen now, I'm going to tell you just the example of differentiation of the muscles from here, but it is very similar to the rest of the organism. So here we have the myotome cells, and the myotome cells are those cells that are determined by the signals of low sonic hedgehog and wind. Okay, you remember low sonic hedgehog, which is this dotted line here, and the that is coming from the dorsal part of the neural tube, and this is important for stimulating the stimulating the transcription downstream of those pathways, the transcription of some genes of the family of MyoDs. MyoDs are genes that are important for the differentiation of the muscle. And they are also master genes, but in that case, like PAX1 is for the cartilage, for the chondrocytes. So here MyoD is a master gene family, are master genes for differentiation of the muscle. And there are two genes, MyoD itself and MIF5, which is also member of the MyoD family of transcription factors. Those transcription factors are very important to trigger the differentiation of those cells, those progenitor cells, to differentiate in muscle. Next, we'll have some FGF signaling, which is going to be important for activating proliferation, and then some other genes, among them another member of the MyoD family of transcription factors, like the myogenin, which is also involved in the organization of the different muscle cells in a kind of lineman, okay? And there is also an organization in which different cell-to-cell contact molecules are going to be activated and are going to be applied here, like fibrinating integrins, caderins, and cell adhesion molecules. Okay. Now, this is a step towards from the determination to the organization of the muscle, and further, if we follow this arrow, what we are going to have is the formation, the start of the formation of the myotube. You know that the muscles are polynucleated cells, so there is fusion. This is a fusion that is in this protein here is very important for the fusion of those cells that were aligned here. And after that, interleukins are going to be very important for the maturation of the myotube. And then at the end, in the mature myotube, this is going to be contractile, and it is a differentiated muscle that is going to be able to execute the function that is characteristic of it. Okay, you will have a seminar on those cells here, which are the satellite cells that are very important. This little cell, you see this little cell here, are cells that are protecting the muscle, and in case of accident, they are going to be involved in repair this myofiber. Okay, so we'll talk about that in seminars. Okay. Now, this is the story. This is the story. What I wanted to do here is a story of one of the cells of this differentiation. So something similar we could apply it to the cartilage and so on. Okay. Now let me finish with a couple of slides to stimulate the curiosity to yourself. One thing I think it is nice for you to know that actually each vertebra that you have in the body has been constructed, has been developed by the contribution of 4 different somites, 2 in one side and 2 in another side. Okay. Now this is what you have in this picture here. This is the neural tube. Actually, this is the spinal cord, and there are going to be some somites. One somite here, the other one is in the other side of the screen, and we don't see it here. Now, this somite in the sclerotome, which is going to develop the vertebra, is going to be divided in 2 different populations of cells. Okay, the yellow population of cell plus the blue anterior population of cell of the other somite, the most posterior somite in that case, in this, are going to meet and construct 1/2 of one vertebra. And the same is going to happen with the next one and the next one. This is what you have here. So this posterior part of this somite is going to meet with the anterior part of the next somite, and both together are going to construct 1/2 of a vertebra. The other 1/2 is constructed from the other somites. And in this drawing here, you can pay attention and we'll see some details of the formation of these structures. The other thing I would like to share with you is the origin of tendons. Tendons develop from, tendons differentiate in order to hold bones together with muscles, right? So exactly, you remember that we have these 2 areas here. This is the sclerotome, this is the myotome, and this is the dermatome, right? Now, Actually, in the interaction between the myotome, which are the muscles that are going to develop the musculature of the vertebra, and the sclerotome that is going to develop vertebra itself, between both here is where the tendons are going to be developed, and some cells will be determined to be the progenitors of the tendons. And this is what you have in this picture here. Those green cells are the cells that are going to develop the tendons. And it is an interaction, so a communication between those two tissues. And this is done because the red cells here, which is the myotome, are sending some signals to the first row of cells of this sclerotome, these green cells. So those signals, which are mainly FGF, and those cells that reach this signal from the red cells, the myotome, are sending the signals to here, and those signals are FGF, and the first row of cells of the sclerotome, which are the ones that receive this signal, are the ones that are going to trigger a particular program of differentiation towards the tendons. And actually, the gene regulatory network that is working in this point here is what you have in this scheme. Okay, this is the myotome. Myotome is sending FGF. FGF is targeting the first row of cells. And this first row of cells that, in the absence of FGF, they would be sclerotome, but now they are just changing the program, and they are activating another transcription factor, which is called the scleraxis, and this guy here is going to develop the tendons. Okay, so that's the way how it is developed here. And actually, if I would like to finish with this part. I would like to finish with this drawing. This is a kind of drawing that you can use for curiosity, and because this is the fate map of a somite, and this is the result. Okay, what is developing. So if you follow the different colors of the different cells here of the somite, you will learn to each part that those cells develop for the vertebra. So each piece of a vertebra, we know exactly what populations of cells of this somite are contributing to. So inside of the sclerotome and inside of the different parts that we have seen here, there are also some compartments, some domains that are going to contribute, and there are a lot of interactions here. Now, I would like very much to stop here and introduce the next set of the next block of nervous system. But if there is any question about what I was telling here and you want to ask it, just ask it, or we can do it later at the end. Okay. So in few minutes, I would like to introduce a few things about development of the nervous system. I'm not going to talk a lot about nervous system because most of the students of the University of Barcelona already studied the development of nervous system in a course on neurobiology. Okay, so I'm just going to the hot topics and some important things. But let me show you this scheme here that corresponds to the ectoderm. And actually, we are talking on the ectoderm, and the ectoderm has to make a very important decision to become nervous system or to become epidermis. Okay, and this is something that is going to be is going to be very important because the lineages are totally different, and actually, as you can see, the epidermis, all these structures here that range from the hair, the nail, the sebaceous glands, and the lens, the cornea, and so on, they are coming from the epidermis layer of cells, and the neural tube is going to develop the brain, the spinal cord, and different retina, and the pituitary, different structures. So the lineages are totally different. Next week, we are going to talk also about the neural crest, which is an area that is just between these two different tissues and develops other structures. Okay, so let me tell you about this decision. I would like to talk today about the decision to be epidermis or decision to be nervous system, and this is something that is intrinsically related to this ectoderm. Now, a typical lesson on development of the nervous system would be to talk about neurulation, development of central nervous system, action, guidance, and wiring. And this is not what I'm going to do here, because I told you that some of students already have it. But you will have also a seminar in the series of seminars in that symposium that we'll organize for yourself. There is a seminar, at least one, that is going to talk about nervous system here, okay? Now, the question is that what I'm going to do is to integrate everything that we had, okay? And we have studied the somites, and now we are talking about the neural tube, and everything that we have studied so far, and put it together and try to understand what is happening here. And the summary, everything that I have to tell you, it is exactly in this movie. So, if you look at this movie, those are embryos. I'm going back to the xenopoof embryo because it's very easy to understand. Okay, so you have the embryo, you can watch it, and you can see the formation of the neural tube, and it is beautiful. If you pay attention to it, you can see here. And this is the neural plate. The neural plate folds and forms the neural tube, the anterior part of the brain, and the spinal cord to the back, and that's it. So the question here that we are trying to understand is how this white zone here, the one that you have, which is the dorsal ectoderm, how do the cells know that they are dorsal ectoderm and they are different from the rest? And actually the answer, you know it already. But first, let me tell you something about anatomy, because this is going to be very helpful for you and for understanding the rest of the story. This is one of those embryos, as we saw in the movie, okay? So you have the embryos here that are folding. Now, so you have them here, and at the beginning we had that white area, which is the neural plate, and this is going to be folding, and then just pushed toward the interior of the embryo. This is going to result in the brain in the anterior part and the neural tube in the posterior part. We have the brain inside, okay? So it has to enter. And then it is covered by the epidermis. Everything it is covered of the epidermis, because we have the epidermis covering all our nervous system, except in two holes in the anterior part, which are exactly the holes where some brain is going out, like the retina or the eyes, right? This is the process. This process is called the neurulation. And if we cut the embryo, if we do a section in this level here, we are going to have something like that. And then progressively, we'll see this neural plate that is folding and finally making a tube. And this tube is just a push in the inner part. Now, if you make a section in that direction, you are going to have this vision here also. So we have the plate here, then there are some folds, and the folds enter inside. Okay, so this is a kind of image that you have to play with it. You take it and you try to understand everything. But let me tell you something very basic. How does this story start? And actually, you know it already, because we worked a little bit this story, right? So we knew that the signals of the BMPs, which are typically ventral signals, they have to be stopped by antagonists on the other side. And this was important for the mesoderm, but also for the ectoderm, okay? So the cells of the ectoderm that stop the BMPs, the action of the BMPs, those cells are going to become the dorsal ectoderm. And the dorsal ectoderm is exactly the neural plate. And the rest is the ventral ectoderm, which is going to develop this area here, is going to develop the epidermis. Therefore, those antagonists that are coming from the organizer are very important, not only to keep the notochord and the somite, but also to keep this part of the nervous system. And this is also valid for the wind signaling, the wind antagonists, like those guys here, Frisbee, Dickoff, and so on, and the BMP antagonists like Noggin, and so on. So this is important for this story as well. Now, then we have something very provocative here. So we know that there is a very important decision to make in the embryo, and it's to be epidermis versus brain. Now, if epidermis needs to be signaled by some molecules, as we saw here, now the question is, what is the default structure for the ectoderm? What is the default structure? Now, is anyone here in the class listening the story that is able to answer to this question? What is the default program of the ectoderm? What do you think? So let's say that you have, it should be the brain in other things because you have it here, right? So, but it should be the brain. But this is very speculative, Laura. This is something that it is very speculative. That the main idea is that if to have if to have epidermis, we need signals. It means that the default is not epidermis. And actually, those guys here, what they're doing is defending themselves from those signals. Okay. Now there is a new proposal that actually the default model is in the absence of cell signaling, the ectodermal cells will adopt the neural fate. And actually, there are some experiments in that. It doesn't mean that if we remove the BMP, that then we'll get a brain on a plate, right? On a petri dish. Okay. Now it is not exactly like that, but some experiments have been done, and it has been found that ectoderm in certain conditions is able to activate some markers of the early nervous system. Okay, so you need indeed some signals to be epidermis, and probably that is telling us that maybe this is a very provocative model. Okay, so this is something that there is a lot of discussion behind that. But there is another thing that I would like to share with you, using the same images that we used at the beginning of the course. Okay, now what is the nervous system? So we were saying that inhibition, those antagonists, inhibition of BMP and X-Wind, so we mean of winds, this is going to be important for determining the dorsal ectoderm. Actually, today we know that low levels of FGF are also important activation. Okay, so there are some low signals that are important for the differentiation of the brain, not only inhibition. Okay, so there is something else than that. Okay, there is something else than the default. So it is more complicated. Okay, now, but in any case, this inhibition is important for having a structure of the ectoderm that is going to be the dorsal ectoderm, and from which the nervous system is going to develop. And the rest of the ectoderm is going to be epidermis, and this is driven by inhibition of those pathways, but also by low levels of FGF. And what is happening during gastrulation? You remember this drawing that we were using for gastrulation? We had here the organizer. This is the notochord. These are 2D structures: endoderm, presumptive head endoderm, and presumptive head mesoderm, and they're the first ones that by involution will reach the other side. Okay, so this is the drawing. This is the drawing that you were studying before, a couple of weeks before, and here we have the positioning of these areas here, okay, in the embryo. This is posterior, this is anterior. But now, look at the picture here. We have something that is very important in those stages, and then is those two regions here are just sending inhibitors of BMP and of Wind to this part of the dorsal ectoderm. And those regions here, which is the notochord, is sending inhibitors of BMP in this part of the ectoderm that is going to be mainly the spinal cord. You can see the drawing, so we still have some problems here, okay? You don't see the drawing at all. Some people say that they see it, some others no. Laura, if I move the picture, you don't see it. Laura, you don't see it? If you don't see it, please say it. I'm going to upload it again, okay? You don't, you see only, okay, I'm going to upload the, and you tell me if you see it, okay? Just, I mean, there are a lot of problems with that. Just wait a second. We have time, okay? We are going to finish in a couple of minutes, okay? So just let me upload it again. Laura, please tell me if now I'm going to play with the cursor, with this pointer. Now you see it? Okay, great. So, okay, thank you very much for telling me. Now, what I was saying here is that actually this is the behavior of the organizer, but then those tissues are entering by involution, and those cells here, the first cells that enter by involution, which is those two, the yellow one, the head endoderm and the head mesoderm, they are the first ones that are reaching the most anterior part, so the head is going to be here. And those guys are sending the inhibitors of BMP and the inhibitors of wind, and this is going to be very important for the anterior part of the nervous system. The anterior part, the brain is the anterior part of the nervous system. Okay, so we are talking about, and this is and the notochord here is going to be sending BMP inhibitors in order to define this area here as the part of the neural plate that is going to develop the spinal cord. Now we have a difference here and about BMPs and X-Wing. In both cases, BMP are acting in order to define the nervous system, but there is a difference to what is going to be the brain from what is going to be the spinal cord, and this is related to the capacity of inhibiting wind or not. Okay, this area here, we call it precaudal plate of the nervous system. Precaudal because it is just anterior to the notochord, and this area here we call it epicaudal. Epi, it means above, okay, so on top, whatever. Okay, so above notochord plate. Okay, so epicaudal and precaudal. Precaudal, pre means in before. Okay, now so this is the notochord, and just the ectoderm on top of the notochord is going to be the epicaudal plate, and the ectoderm on top of this mesoderm, head mesoderm, and head endoderm and head mesoderm is going to be the precaudal plate, and both are the neural plate. So if you allow me to go back to the movie, okay? Now, oops, I hope you see the movie. No, okay. Now this was the plate, okay. So here we go. It is here. Now I'm going to stop it when you see it. Oh, okay. So that's the plate, the neural plate. This corresponds more or less, approximately. Okay. So this is the epicaudal plate, and this one here is the precaudal plate, right? And if we go to the movie, this would correspond more or less to the precaudal, and this is the epicaudal plate, okay? More or less, okay? So that's just to orient. Now, what I'm saying that, and what I want you to show here is that if we just remove from that embryo this area, and this is something that is very similar to what happens in mammals as well. Okay, so we have here the precaudal plate, and this is the epicaudal, and this is the notochord, and this is this head endoderm and mesoderm, and those guys here are sending inhibitors of BMP and inhibitors of wind, and those guys here, the cells of the notochord, are sending inhibitors of BMP, but also they are sending, there is some wind in later stages that is produced here in order to determine the posterior part of the brain. So there is also some retinoic acid and some low levels of FGF that are acting here. I mean, the story is a little bit organized. So my last slide, the message that I want you to take home is what you have here. Now, that early decision that the ectoderm has to do to be epidermis or to be nervous system is exactly what is depicted here by a signaling mechanism. If there is BMP, it is going to be epidermis. If there is no BMP, the chances is that it's going to be brain. Now, if that BMP to be epidermis will need some wind, okay? Now, in the absence of BMP, in the absence of signal, the default model is going to be nervous system. If there is a bit of wind, and also low FGF and RAS, this is something very new that has been discovered not many years ago. Okay, so if in addition there is some wind, this is going to be the trunk, the spinal cord of the nervous system. Okay, now if there is no BMP, no wind, this is going to be some head or some brain, so the most anterior part of the nervous system. Okay, and with this image, I would like to stop here, and that's all I wanted to tell you. And thank you for waiting a bit longer than what was programmed this talk. And just let me ask you a question. So since I switched off the image, the video, could you hear it better, or still there are some problems? I'm trying. Do you think it's better, Antoni? So, any other opinion? There was no voice interruption. So probably this is what we'll do exactly. So this is probably what we'll do in the other class. And I'm sorry about that. You, Valentina, you had voice interruption. I had a couple of issues, but I think it's due to my connection. Valentina, that was at the beginning, before I closed the video, or after? It's important to know it for us, okay? Because we have to detect before and also after. Okay, some people video was closed at 3:52. Nothing changed. Okay, some people. Valentina, are you in Barcelona? Yes, okay. That's what I thought. Marta told me. Marta comes. So we probably have something. I don't know. For some people it went better. This is the conclusion. For others, did not change. In any case, what I'm trying is.